Hello, I'm Ayse Dincer. I'm doing my PhD in computer science at the University of Washington. Today, I'm going to provide an overview of my new and ongoing project where we infer peptide coefficients from quantitative mass spectrometry data. Tandem mass spectrometry, or MSMS, can be used to quantify thousands of peptides in a complex biological mixture. However, the problem is that these measurements depend in part on the properties of the peptide sequence. These properties can be how susceptible the peptide is to enzymatic cleavage, or how efficiently it traverses the liquid chromatography column, or how easily and uniformly the peptide fragments in the dissociation phase. Our goal in this project is to train a machine learning model to quantify these peptide-specific properties, and then adjust the observed quantities to remove these effects. Our end goal is to offer our approach as a pre-processing step to eliminate peptide bias from quantitative MSMS data, so that these adjusted quantities can be used for a wide range of quantitative MSMS analysis. Our approach rests on two primary assumptions. The first hypothesis is that peptide measurements can be decomposed. Here we have a schematic of our matrix Q of peptide measurements where we denote one cell as QIK, which is the observed abundance of peptide I in run K. Our hypothesis 1 states that QIK can be decomposed into a peptide coefficient Ci and an adjusted peptide abundance alpha Ik. So the Ci here is the quantification of the peptide-specific effects we just talked about, and alpha is the adjusted abundance that we want to reveal. We build our second assumption on the first one and assume that unique sibling peptides should have equal adjusted abundances within a single MSMS run. By unique sibling peptides, we mean peptides that occur exactly once in the protein database and that co-occur on a given protein. So our hypothesis 2 states that for run K and sibling peptides I and J, alpha Ik should be equal to alpha Jk, which are the adjusted abundances. So the key idea here is that if these peptides occur once, and if they occur on the same protein, then their measurements should be the same. So our goal is to infer coefficients Ci and Cj such that we make the adjusted abundances equal. We use these two assumptions to infer peptide coefficients from the peptide measurements to reveal the adjusted abundances. Accordingly, we first provide empirical evidence for the consistency of these sibling peptide intensities. Second, we implement a neural network to predict peptide coefficients from the amino acid sequences of the peptides. And then we show the ability of this neural network to make predictions for new runs and new proteins. Let's start with the first experiment. Before delving into the details, for this and the remaining experiments, we analyzed quantitative MSMS data from human cerebrospinal fluid. Each individual sample in our dataset was collected by data-independent acquisition, DIA. Also, the intensity measurements in the Q matrix denote the summed fragment ion intensities of the peptides. And we also eliminated all shared peptides from our dataset. And by shared peptides, we mean peptides that appear in more than one protein. Our goal is to investigate the consistency of intensities of sibling peptides across multiple runs. So for this, we randomly select from our Q matrix a pair of unique peptides Pi and Pj, and a random pair of runs Rk and Rl. Based on our hypothesis, for sibling peptides, we expect the ratio of intensities of peptide I in runs K and L to be approximately equal to the ratio of intensities of peptide J in the same two runs. And this simply follows from the assumption that adjusted abundances of sibling peptides should be equal in one run. But the key thing here is that we do not expect to observe any correspondence between ratios of non-sibling peptide pairs. In this plot, we see the distribution of the ratios of abundances for 10,000 randomly selected non-sibling peptide pairs. So the x and y axes denotes the ratios for peptides I and J. And as expected, for the non-sibling peptides, we observe quite a random distribution. But when we created the same plot, this time for sibling peptide pairs, we observe that the distribution is quite along the x equals y line. So this result indicates that the intensity ratios of sibling peptides are quite consistent across multiple MSMS runs, just as we hypothesized. After this empirical investigation of the sibling peptide intensities, we now move on to defining our neural network, which will take a peptide sequence and predict a peptide coefficient. Let's first start by defining our loss function for our neural network. But first, we make a change of variable. Instead of inferring coefficient Ci, we will infer the multiplicative inverse Di, which is really important for avoiding numerical instability issues. Our loss is based on the assumption that the adjusted abundances of sibling peptides should be equal. So for all pairs of sibling peptides I and J, we're trying to minimize the distance between QIKDI and QJKDJ. So since we made the change of variable, you would notice that this is simply the adjusted abundances. And since we're calculating the loss in log space, we're effectively trying to make the ratio of adjusted abundances of sibling peptides 1. And of course, we sum this peptide loss over all runs K in our datasets and over all proteins. We also have two regularization losses. The first term is trying to make the sum of 1 minus Di values across all unique peptides 0. And the second is L2 regularization on 1 minus Di values. So these two losses are for the proper scaling of the coefficients. We want Di values to be 1 on average, but we also want to avoid making them too large or too small. And of course, we have the constraint that all D values should be greater than 0. Now let's move on to defining our neural network to minimize this loss. Our goal is to define a coefficient predictor network that takes a one-hot encoded peptide sequence and outputs a coefficient Di. But a key point is that our loss function is actually a series of independent loss functions for each protein. So this means that the coefficients we learn for one protein are not necessarily calibrated relative to the coefficients learned for a second protein. So to solve this problem, we adopt a Siamese architecture. What this means is that we have two copies of this coefficient predictor network S with the exact same architecture and weights. And each arm of this network takes a peptide sequence and outputs a coefficient. And we train the entire network using all pairs of sibling peptides in our dataset. And one key point here is that even though we train our network from only the unique peptides, the coefficient predictor network can be used to make predictions for shared peptides as well. Our coefficient predictor network S consists of convolutional layers to extract k-mers from the one-hot encoded peptide sequence and also dense layers to finally predict a coefficient Di. When we experimented with different architectures, we observed that adding these convolutional layers can improve the model performance. Now that we have defined our neural network model, let's move on to predicting coefficients for neurons and new proteins. We started with the task of predicting for neurons. For this, we subdivided our Q matrix into a train and test set of runs, trained our neural network from the train matrix, and made predictions for all peptides. Here you see the distribution of the observed abundance ratios for the train matrix. So in other words, for all pairs of sibling peptides I and J, we plotted QIK divided by QJK. And we, of course, created the same plot for the test runs as well. So for both sets, we see that the original distributions show quite high variance. But then, we adjusted the abundances in the train run by multiplying them with the predicted DI and DJ values. So, in other words, this time we created the distribution of adjusted abundance ratios. And as expected, we see that the distribution becomes more peaked at 1, which means that the ratio of adjusted abundances become closer to 1 for many pairs, which is exactly what our loss function is trying to achieve. But the key point here is that when we also adjust the abundances of the peptides for the test set of runs, we again see that we managed to reduce the variance in the distribution. But we also wanted to quantitatively investigate these results. So we first calculated the mean squared observed abundance peptide loss, which is the log squared distance between QIK and QJK for all pairs of sibling peptides. And then we adjust the abundances with the predicted coefficients and calculate the mean squared peptide loss of adjusted abundances. When we calculated these two losses for the train and test sets, we observe that we can successfully reduce the loss by almost 50% for both sets, which indicates that our neural network can predict coefficients generalizable across runs. Finally, we moved on to the key and most challenging task, predicting for new peptides occurring in new proteins. For this experiment, we first split all the unique proteins in our datasets into train and test runs, and accordingly defined the set of all train peptides occurring in train proteins and test peptides occurring in test proteins. We also split our runs into train and test sets so that our test matrix consists of a disjoint set of peptides and runs. And we trained our neural network from the train matrix and predicted coefficients for train and test peptides. Here we're comparing the distributions of observed abundance ratios and adjusted abundance ratios for the train set. As expected, we see that we managed to reduce the variance in the distribution and also successfully reduce the mean squared peptide loss by more than 50%. But the more interesting part is that when we created the same plots, this time for the test set of peptides and runs, we observe that we can again make the distribution of adjusted abundance ratios more peaked at 1 compared to the observed abundance ratios, and also again reduce the test loss by almost 30%. This is a promising result because we're passing the test peptide sequences to our neural network and using the predicted coefficients to successfully adjust the abundances in a test set of runs. So these results show that our neural network can make accurate predictions for new sets of proteins and runs. Looking ahead, we want to investigate transferring our model across datasets. And we see pre-training our neural network on multiple datasets and then fine-tuning on the dataset of interest to be a promising future direction. And once we're successfully transferring across datasets as well, we want to investigate whether our approach can improve the accuracy of fold change measurements. And basically, we want to further explore the extent which our approach can be useful. So our goal is to offer our neural network approach as a preprocessing tool for quantitative mass spectrometry data. And so we want to carry out experiments to examine how these adjusted abundances can be used for various quantitative MSMS analysis. Before concluding, I want to thank my advisor Bill and my co-authors Jan and Sriram, and also all members of the Noble Lab. We're very excited about this project, and I hope it was interesting for you as well. Thank you for listening.